Hello, everybody. My name is Lee Brown, and as Gary said, I'm based in the U.S. I'm actually in Texas, and Bio-Rad as a company is in California, but I cover basically a third of the U.S., which is a little bit nutty, but my main role is helping instructors bring these kind of labs into their classrooms and working with curriculum, helping people figure out how this is going to look in their classroom. And so that's what we're going to talk about today, really, is how to prep this lab, how to make it work in your class. We could spend hours and hours talking about CRISPR, and I didn't put a lot of CRISPR background information in here, but I do want you to know that that is available on our website, and I think Gary's going to link it as well, or he said he could do that later on. So if we don't have it up now, then I'll give you the links in here. We're focusing on this out of the blue CRISPR Gene Editing Kit, and this kit is kind of self-contained in a box. It has everything you need except for the equipment, and we'll talk more about the equipment in a minute, but it has all of the plastics and reagents that you need in order to do this with your students. We typically have our kits parceled out for 8 lab groups, working in groups of 4, and so 32 students total. That's not to say that, you couldn't have 3 people per group. If your classroom is not 32 students, you're lucky enough to have a smaller class, then you're more than welcome to, have them work in groups that are a little bit smaller. But yeah, 8 lab groups and all of the plastics and consumables you need there. So I'm not going to, like I said, I'm not going to go too much into how CRISPR works, although I do have slides for that ready. So if during the Q&A you want some more background, then we have time, then I'm happy to go into that. But the big overview here that I want to get to is that in this lab, your students are going to start with blue colonies, and those colonies are expressing LAC-Z. So the LAC-Z gene makes these colonies turn blue. I'll show you a slide on that in a minute. They are going to do CRISPR gene editing, and that gene edit is going to knock out the LAC-Z gene, so the colonies are now white. Okay, so we're going from blue colonies to white colonies. Okay, and so here's kind of how that looks. So with functional LAC-Z, we have that bacteria, the E. coli, with a functional LAC-Z gene is making an enzyme called beta-galactosidase, or beta-gal. And beta-gal usually works on lactose, but we're giving it an artificial substrate called X-gal. So X-gal is kind of like lactose. Beta-gal will break down that artificial substrate and make a blue pigment. Okay, and so if LAC-Z is functional, then our bacteria will be blue, like this picture on the right shows. Okay, now if we have LAC-Z that is broken, so let's say we add a stop codon in there, which is what we're going to be doing in this lab, that means that LAC-Z will not be transcribed and translated. We won't make beta-gal. And so therefore, even if they're on plates that contain X-gal, they will not be able to break down that blue pigment, and those bacteria will be white, right? Or this creamy color that you're probably kind of used to seeing E. coli. And again, these 2 bacteria are growing on the same exact plates. It's just that the white bacteria are not, do not have a functional LAC-Z gene because it's been edited. Okay. Now, the lab itself is a transformation lab. So if you've done PGLO, then this should look pretty familiar, where you have your starter plates, you're adding a plasmid, you do a heat shock, and then they're plating their bacteria. I have several people who are curious to see if they could use this instead of PGLO. And yes, in theory, you could. The technique is very simple. We have in the US, we even have middle schools that is, age 11, 12 doing this lab. The science behind CRISPR is a lot higher level though. And so having them, get to the point where they can understand the science might be a little bit tricky. I really recommend people do PGLO first because that lab teaches them what a transformation is. The idea that, bacteria have small circular pieces of DNA, and we can get those, we can change that DNA to have genes that we want. And then we can use this heat shock protocol to get the DNA into bacteria. And then we can use antibiotic selection to select the bacteria that got the plasmid, right? And so that's all kind of covered in PGLO. For the CRISPR lab, even though we're using a bacterial transformation, we kind of assume that they understand how transformation works, what a plasmid is, how antibiotic selection works, all of that. And so I kind of compare it to, how when students are little, 5 years old or, up to 8 or so, 7 or 8, they're learning how to read. And then once they get older, they're reading to learn, right? And so in this lab, we're taking a technique that they learned how to do with PGLO, and we're applying it to this skill of CRISPR-Cas9 gene editing, okay? So not to say you can't teach transformation with this, you're just going to need to slow it down quite a bit and add in all those supports, okay? So just as a big picture look here, CRISPR gene editing has two basic parts, and you've probably heard of CRISPR-Cas9. Cas9 is the enzyme that does the cutting, and CRISPR is really named for some of the components here. When we discovered this, it's clustered regularly interspace palindromic repeats, and that's a mouthful, but basically it's a stretch of DNA. And so there is Cas9, and that DNA is transcribed into an RNA, and this RNA is actually hooked up to a kind of the scaffold piece, this orange piece right here. And together, this is a single guide RNA, and that single guide RNA has this blue target region. And Cas9 and the single guide RNA kind of move as one unit, and they scan DNA, and they're looking for a match with this target DNA here. I'm sorry, with this this guiding region. I said target earlier. This guiding region of the single guide RNA is looking for a match with the target DNA. So you can see the blue nucleotides up here are the match. And when it finds that, it makes a double-stranded break. Okay, and so this is like I said, the Cas9 itself is a nuclease. It cuts the DNA. It's not used up. It's free to go and go on to other pieces of DNA and cut it then too. And so we have a cut in the DNA. Now that is the cutting action of CRISPR, and that alone would knock out Black Z, but we're going to talk about something that's kind of cool in this lab. So the real power of CRISPR is not just making the cut, although that is extremely powerful. The fact that scientists can kind of program in this guiding region on the single guide RNA, you can just order these up, right, from different suppliers and say, I want this sequence because I found my gene, I found the cystic fibrosis gene, for example, and I want it to target right in this area. And so you give them 20 nucleotides and they'll ship you the single guide RNA, which is really cool. So that's a way for us to target specific genes and cut them, kind of like a restriction endonuclease does, right? Restriction enzyme does. However, we can tell it what our target area is. But the real power in CRISPR comes from the ability to do repairs. And so double-stranded breaks are typically lethal, right, in most organisms. So that's not a good situation. So there's two different repair pathways. I'm not going to go too much into that, but the one that we're looking at is this homology-directed repair. So if you give these organisms, which have the cut DNA, a piece of donor DNA, which is shown right here. So here's our cut DNA at the top. And if you give them a little piece of donor DNA that matches up with the cut, arms, like the flanking ends here, it will incorporate that into the cut piece, and now you have inserted whatever you want to insert there, right? So you might change bases this way. You might insert the correct base if we've had a mutation. You could knock things out by introducing a frameshift mutation. You could introduce a restriction site, right? If you were trying to get this so where you could cut it later with a restriction enzyme, you could introduce that here. In this lab, we're introducing a stop codon. Okay, and so that piece of DNA is inserted into the cut DNA with the help of these repair proteins, and that's just represented here by this orange blob with a gear on it. Okay, so let's take a look at all these pieces and parts in this particular lab. So in this lab, as I mentioned before, we're targeting the LAC-Z gene, and we need some things in our lab protocol so that we can do all these steps, right? Obviously, we need Cas9. That's one thing. We need a single guide RNA, right? So we've engineered that. We need the donor DNA, okay? So that donor DNA in our example is going to have a stop codon. And we also need those repair proteins, okay? And of course, the target DNA is in the bacteria already. E. coli are expressing that LAC-Z gene typically, okay? All right, so 4 components: Cas9, single guide RNA, donor DNA, and DNA repair proteins. Where are all of those pieces and parts in this lab? Well, all the starter bacteria are expressing Cas9 already. We've engineered them that way, okay? And so they are expressing Cas9. And then we have 2 different plasmids. Okay, so this might get a little bit overwhelming. This is why I say, it's better to probably do pGlo first. So this isn't their first time seeing a plasmid, but I'm just going to briefly go over what this plasmid is, what we're looking at here. And you'll notice that this says PLZ donor guide, or we call this plasmid PDG. You can name this plasmid whatever you want. You can call them plasmid 1 and plasmid 2. Doesn't matter to us in the least. But this plasmid that we've named PLZ donor guide, has an origin of replication that just allows bacteria to make more and more copies of the plasmid so they can pass it on when they divide. It has a spectinomycin resistance gene. So that means that bacteria that take in this plasmid will be able to grow on plates that contain that antibiotic, spectinomycin, okay? So that's the way that we select for the bacteria that were transformed, okay? The next thing that it has is the donor DNA. So remember I said that piece of donor DNA, you can give it to the bacteria and they'll incorporate that into the cut target DNA, right? In our case, the LAC-Z gene. This plasmid itself is the donor DNA, right? So the DNA is just right on there. There's a stop codon encoded in there, and it will be inserted into that cut DNA. And then finally, this area right here is the guide RNA region. And this DNA will be transcribed into the single guide RNA. So that's where our single guide RNA comes from, right? So this plasmid is giving us both our donor DNA and the region that will be transcribed into the single guide RNA. Okay. I'm going to let that sink in for just a second. And like I said, if you have questions, just pop them in the chat and Gary can let me know. Okay, so that's one of our plasmids. Now, this is going to be our plasmid that has all the pieces and parts that we need, right? But we want to show students what happens if we don't give it a single guide RNA. So we have another plasmid, right? And this one looks exactly the same. Origin of replication here, spectinomycin resistance gene here in green. We have the donor DNA here in blue, but we do not have that orange piece that can be transcribed into the single guide RNA. If we transform bacteria with this plasmid, they will not be expressing that single guide RNA, and so Cas9 won't know where to cut. You know, the single guide RNA is kind of like the GPS for Cas9. Cas9 otherwise is just driving and puttering around in the cell, you know, kind of aimlessly. The single guide RNA is what's telling it where to go and what to cut, right? And so without that, Cas9 won't cut anything. Okay, so this is one of our controls. See, kind of helping students understand what that single guide RNA does. Okay, so let's break that down in a little bit more graphical form here. So we have our PLZ donor guide. The DG is the donor guide, right? The guide is the real piece that we're looking for here. It does have the single guide RNA. That's what sgRNA stands for, right? And the other plasmid does not have that, okay? And so then let's look at the donor DNA. And if you remember right, they both have the donor DNA. They both have that region, I think it was blue, that is the donor DNA that will be inserted, and that's the one that has a stop codon, okay? So the only difference in these two plasmids is one makes single guide RNA and one does not, okay? But they both contain the donor DNA. Now, the other thing that we need are the DNA repair proteins. And those DNA repair proteins are under the control of an arabinose inducible promoter. And that's a mouthful. And again, hopefully you've had PGLO or something under your belt, or you understand how bacteria can be induced using different ingredients in the media, right? Different components in the media. So arabinose is just a sugar. And normally these repair protein genes are turned off. But if we give it the sugar in the media, it will turn those repair proteins on. Okay. And so by using that sugar, arabinose, with or without, we can either decide if the repair proteins are going to be turned on or if they're not going to be turned on. Right. And so there are two different starter plates that you give students. One of them has the arabinose. And so those bacteria are making the repair proteins. The other one is not having the arabinose. And so those bacteria do not make the repair proteins. Okay. And so again, this is another control so students can see, okay, we do need those repair proteins in order to fix that cut, right? So we're trying to get that little piece of DNA to go into our cut DNA, right? And so here's our experimental setup. They'll have an X plate. So this, I'll explain what these mean, but this does not have arabinose, without arabinose, and they'll mix it with those two plasmids, and then they'll do the Xera plate with arabinose and mix it with those two plasmids, right? And so they'll have 4 plates at the end, and we'll walk through the predictions of what they would expect here, okay? Will we get the target DNA cut? And if it is cut, will it be repaired, right? And then what phenotype will we expect to see, okay? So something like that. There we go. So here's all the pieces and parts. And Xera, those bacteria are both expressing Cas9, right? And the X one is not expressing the repair proteins. Xera is. PD plasmid, remember, does not have the single guide RNA. PDG does have the guide RNA, okay? And they both have the donor DNA. So that's kind of this, I have this worksheet in here that you can print out. I think I've got it actually formatted for a regular letter size sheet of paper, and students can make their predictions on here. And so that works out really nice to have it visually like this. Okay, and so let's just go through one plate here. So we're going to look at basically our experimental plate, right? So this is D, and we are in D, you can see right here, it's this one, right? So we're looking at the Xera plate combined with the PDG plasmid. Okay, so here that is right here. And we are looking for all these pieces and parts, right? So do they have Cas9? Yes, they do. All the bacteria are expressing Cas9. Great. So now with Cas9, remember, it won't cut unless it has a single guide RNA, right? So does it have the single guide RNA? Well, we're using the PDG plasmid. Remember that G stands for guide. So yes, indeed, it does have the single guide RNA. So those two components are all you need for a cut, right? So we do have a cut. Great. Check. Done. Okay. Now, remember that cut's lethal, right? So bacteria don't like to have their DNA cut because everything downstream of that gets messed up, okay? So we want to fix it. And in this case, we want to add a stop codon in LAC-Z. So we need the donor DNA to do that. Now, remember, the donor DNA is in both the plasmids, but it is in this PDG plasmid, obviously, right? Here's our donor DNA. But we need the proteins that are going to stitch that back into the cut DNA. And since these bacteria are grown on arabinose, they are indeed making those repair proteins, right? And so that repair can happen, and that happens pretty much instantly, right? Like in the tube before you plate it even. So that repair happens, and now we have our LAC-Z gene repaired, but it has a stop codon in the middle of it, okay? So now let's look at that kind of phenotype again. Whoops. And you can see, what phenotype would you predict from D? Okay, so we have our blue colonies here, our white colonies here, or no colonies, right? So this will be our lethal phenotype. This would be a phenotype that we would expect to see if, for example, Cas9 did not cut LAC-Z, right? And this would be the phenotype that we'd expect to see if Cas9 cut LAC-Z and repaired it using that stop codon, right? So this is the phenotype that we would expect from D, these white colonies. And hopefully that makes sense. All right. And then they would go through and make some predictions on these other ones. And I'm just going to fill this in for you here and quickly explain it just in the interest of time. Normally I, have you guys talking about this in the chat and whatever, but just in the interest of time, we'll go through this quickly. So in the case of A and C, both of these are going to be very similar. We've got Cas9, but we don't have a guide RNA. So without the guide RNA, Cas9 doesn't know where to cut. So LAC-Z is going to be intact, and we're just going to have blue bacteria, just like we did before, right? Now, I will say, you might be wondering, well, why are they growing on this plate? Or why don't we see a lot of them? Remember, we're only allowing the bacteria that took up the plasmid to grow. These plates contain that antibiotic spectinomycin. And so these bacteria actually were transformed. They do have the plasmid in them. It's just that their plasmid does not have the directions for making that single guide RNA, okay? It does, however, have the gene to allow them to have spectinomycin resistance. So that's why they're growing on this plate. Okay, so those are blue colonies we would expect in A and C. And again, it's because they're using the same plasmid, and that plasmid does not have that single guide RNA. Now, let's look at B. B is really a cool case. So B, we have Cas9 right here. We have the single guide RNA, so we get a cut, okay? Now, we've given it the donor DNA. That's on the plasmid itself. Oops, right here. But these bacteria are not making the repair proteins. So even though it's got all the parts here, it doesn't have a way to put that donor DNA into the cut LAC-Z gene. So now we're left with bacteria that has cut LAC-Z and no way to repair it. And that is a lethal phenotype. And so what we see in this particular I've lost my cursor here. There it is. What we see in this particular phenotype is no bacteria on their plates at all. Okay. And so that's a good one for having them think through this process of all the things that are necessary for this gene editing to occur, right? The cutting and the repairing. Okay. Now, C we talked about already. D we've talked about already. D has all the components. We have Cas9. We have the single guide RNA. So we get a cut. We have the donor DNA, so we get the repair, and we have the repair proteins right here. And so we get the white colonies here. Okay. And so this will take them a bit of time to kind of walk through and argue about and all that good stuff. But that's a really valuable lesson for them. So let's quickly go over the protocol, and then I want to get really into the prep here. So big picture, they're combining the 2 starter bacteria with the 2 different plasmids. They're going to do a heat shock, and that's going to get the plasmid DNA into the bacteria. And then they'll plate the bacteria, and that gives us a way to select which bacteria took in that plasmid. Remember, again, the plasmid has that gene for spectinomycin resistance, and so only bacteria that have that gene will be able to grow on these plates. These plates all have spectinomycin in them. Okay, so again, another difference from PGLO. PGLO, you're dealing with 3 different types of plates here. This lab, all these plates are the exact same. We're just plating different things on top of them. Okay. So they're going to start off by labeling their tubes and putting them on ice. And then they'll add transformation solution to their tubes. Okay, so 250 microliters of transformation solution. They can use a P1000 pipette for this, or if you have disposable pipettes, they could do that. Disposable pipettes don't come in the kit, but you know, usually you have these laying around, so there you can use those, and you can even wash and reuse those. But this would be, you know, the transformations. Well, I don't know if I'd wash them actually. We're dealing with bacteria. Probably a sterile one would be best in this case. So the 250 microliters from either a micropipette or a disposable transfer pipette. Then they're going to add the X bacteria, the bacteria growing on this X plate. This stands for IPGG and XGAL, and I'll talk more about that in a minute when we talk about the prep. But from this plate that does not have the arabinose, right? See, it's just X, no X era. They're going to take 5 colonies and put them in tube A, and they'll swirl their loop around, you know, do the little washing machine action with their loop. I like to tell them for a minute. We want to make sure that that bacteria is really good and dispersed in the transformation solution. And so they're going to, you know, just kind of do their hands like this and make sure that there's no clumps of bacteria in that solution whatsoever. Okay. It should look evenly bluish, right? Basically, they'll use a new loop and do it for tube B. So A and B both get the bacteria from the X plate. Now they're done with that X plate, and they're going to move on to the X era plate, and that bacteria will go into tube C and D. Same procedure. Pick up 5 colonies, add it to tube C, swirl it around in there for a minute or so, get a new loop, and do it in tube D. Okay. So now our tubes have transformation solution and they have bacteria, right? And A and B have one kind of bacteria, C and D have another kind of bacteria. Next step, they're going to use a micropipette to transfer 10 microliters of PD plasmid. Okay, this PD, this is the one that does not have the single guide RNA being expressed. Okay, into tubes A and C, right? So they're going to need to be careful here which tubes they put it into. 10 microliters into tubes A and C. So they'll need a P20 micropipette for this step. I do have alternatives for you if you don't have pipettes. And then they'll, and they want to switch tips between these tubes, obviously, so using good sterile technique. I usually have them put the tip into the solution at the bottom and then depress the plunger on the micropipette. And then I like to have them pipette up and down a couple of times, right? Just to make sure they're getting it all out of the tip and mixing it up. And then they'll use a new tip to put more plasmid into tube C. They'll do the same thing. So again, switch tips, of course, and use the PDG plasmid. And PDG will go into tubes B and D. Okay. And so those are going to go on ice for 10 minutes. And during this time, this is when we're doing kind of this cold treatment on the bacteria. Their cell walls will, you know, the cell membrane will slow way down. And we want them good and cold before we do the heat shock, okay? When they do the heat shock, this can happen in a water bath at 60 degrees Celsius. You'll notice that this is a bit higher than 42 is what we typically call for P-Glow. We found that 60 degrees, 42 just flat out didn't work. This is why we have our R&D scientists in the lab. And so she kept kind of increasing the temperature, increasing. And it turned out that 60 degrees was a sweet spot. And so we do a heat shock for 60 degrees Celsius for 50 seconds exactly. Just a side note here, if you do P-Glow at this temperature, you will probably see increased transformation efficiency as well. Okay, so she did try it with P-Glow and saw that that actually worked a little better. Okay, and it's probably comes down to the fact that the heat getting into the tube, it takes a little bit of time for that to get up to 60 degrees into the tube, right? It's probably not even getting up anywhere close to that, but that 60-degree help transformation efficiency quite a bit. Okay, so they'll go from the ice straight into the water bath. You could use a dry bath for this if you prefer, 50 seconds, and then straight back into the ice. Okay, and some tips here, the tubes need to be pushed through the float so that the bottoms of the tubes are actually making contact with the water. That's really important. And they need to, we don't want them taking it out of their ice bucket or ice cup and then walking over to the water bath. We want them taking their ice bucket to the water bath so they can transfer it and put it right back in ice right away. So we want that to be quick. Now, during that heat shock, that's when some of the bacteria are going to get the plasmid kind of getting in through some of the pores that open up when the bacterial membranes start becoming more mobile. And so only, this might be 1 out of a million bacteria or less, right? But some of those bacteria will be lucky enough to get the plasmid. And I say lucky because they're the only ones that are going to survive our next step. They will be starting to express the genes, especially for spectinomycin resistance, right? And so that's what we want to happen. Okay, so that's the heat shock. This is a really important part where the plasmid gets into the bacteria. They're going to stay on ice for 2 minutes. That's just to kind of close that cell membrane back up. And then we're going to let them recover. We're going to give them LB broth. And this is just LB is just Luria broth. It's, got it's chicken noodle soup for bacteria, right? Got all the goodies, amino acids, nutrients that bacteria like. And so we give them that. All the tubes get 250 microliters. Those stay at room temperature for 20 minutes. I prefer, if I've got an incubator there already set, I prefer to just let them recover at 37 because that's the temperature that they really like. If you don't have an incubator, that's fine. If students want to hold them in their hot little hands, that's great too, right? That'll be a little bit warmer than room temperature. We're just trying to be really nice to them now, right? We've put them through a lot. And we've, frozen them. They don't like being at 0 degrees, and they definitely don't like being at 60 degrees. Some of them will die in that whole process, but the ones that survive, the real troopers, we're going to baby them a little bit here. So give them that broth, let them recover at room temperature for 20 minutes. This is a good stopping point. If you are on a 45, 50-minute schedule, then you can stop and leave the tubes at room temperature overnight. Do not put them in the fridge. Just leave them at room temperature overnight and then come back the next day and plate the bacteria. Okay. If you have longer class periods, so in the US, some of our class periods are 45, 50 minutes, some of them are an hour and a half. It just depends on how the school structures things. If you have more, or if you're in a university and you have a longer lab period, go ahead and go on to the next step. So, and, again, I like them thinking about during that 20-minute recovery, if you have time to go on, have them do this little exercise here with the predictions. Okay. Now, if you have time for them to go on, or whenever they are ready to go on, they are going to have 4 plates that are XSBT. Again, I'll explain that when we talk about the prep. This is just IPTG, XCal, and spectinomycin. And they will label those A, B, C, and D. And then when they're ready to plate these after that 20 minutes or overnight has passed, they'll take 100 microliters from A. They kind of flick the tube to make sure that the bacteria hasn't sunk to the bottom, resuspend it. 100 microliters from A onto the A plate just in the middle, and then they'll use a loop to spread it all over, right? We want them to, I always tell them, try to completely cover that agar surface with your bacteria. They'll be able to see it because it's shiny, right? The liquid's shiny. And then they'll just repeat that process for B, C, and D, right? B goes on the B plate, C goes on C plate, D goes on D plate, so on and so forth. Okay. Oh, and I said repeat step 17 and 18. I need to change that. Apologies. Once they've got their 4 plates labeled A, B, C, D, they're going to let the bacteria sit for a minute and soak into the plate. If they're super wet, we don't want them flipping them upside down and having it drip onto the lid. But once it's kind of soaked in a little bit, it might take a minute or 5 minutes. They're going to stack the 4 plates up and tape them and then they'll incubate them upside down. That means agar side up, right? So we don't want condensation falling on the bacteria. And they'll incubate them in an incubator at 37C for overnight, 24 hours. If you don't have an incubator, that's okay. You would let them sit 2 to 3 days. Sometimes I see colonies in 2 days. 3 days is probably safer though at room temperature. These guys have a lot going on, so you got to give them a little breathing room. The really important thing here is this media is light sensitive, and so you have to keep it in the dark. So if you're not incubating in an incubator, just stick them in a box or in a cabinet somewhere. Doesn't really matter, no problem. If you are incubating in an incubator, I typically put aluminum foil over the glass window here so that it's covered and dark inside there. Okay. Um, Now, for those of you who don't have pipettes, I'm going to show you what you can do instead. We really do recommend a P20 pipette. That's the one that goes from 2 to 20 microliters, just for adding that plasmid. But if you don't have the P20 micropipettes, you can pre-dispense, pre-aliquot the plasmids into tubes A, B, C, and D, right? And that way that you would label the tubes A, B, C, and D and put the appropriate plasmids. So in other words, the PD plasmid goes in A and C, and the PDG plasmid goes in B and D. And then you would give the students the tubes. I like to have them take a look at it. Can you see that little bitty drop at the bottom of the tube? That's your plasmid, right? That's the plasmid. And then they would have the transformation solution on top of that and the bacteria on top of that, right? And now they've got all the parts in there, and you don't have to have micropipettes to do this lab. Okay. This is the way that I do it in workshops, actually, because it gets to be a lot for people who've never used a micropipette to suddenly be faced with 3 different sizes and different tip boxes and all that. And so I typically always just do it this way. And here's how I kind of make the protocol a little bit easier for them to understand. So the one I gave you kind of mimics the one in the manual, but this one works great for putting up on the projector so students can see it. They're going to add transformation solution to all their tubes. Then they'll add the bacteria. So 5 colonies from the X plate into A and B, 5 colonies from the X era plate into C and D, and they'll add the plasmid, right? So that's it. I usually give them the P20 so that they can add the 10 microliters of PD or PDG. Now, again, if you do this for them, you can just circle this and say, we've already put that in there. So no worries there. Okay, so I'm going to stop here and see if there's any questions so far, because then we're going to get into the prep.
Hi Lee, there's been a few questions, but I think me and Donna have been dealing with them.
Okay.
Started off there, someone's asking what the Cas9 gene was, and they're asking what temperature they can have their water bath in the prep room, but I think we're okay to carry on.
Wonderful. Okay, great. All right. So, and I will tell you all that this is a lot of information. So if you're feeling a little bit overwhelmed right now, you are not alone, right? The first time I read through this lab, it took me a while to let everything sink in, and I've kind of recreated a lot of the figures and things just to make it a little bit more clear to me. So the things that you see in this PowerPoint won't necessarily be looking exactly like they do in the manual. And so you can use both of those resources, of course. This is just a way that helped me kind of understand what was going on a little bit better. Okay, so now we're ready to talk more about well, let's look at the results first. So what will you see? So here's some actual experiments that we've done in workshops. So these are results from different participants. So our A and C, remember A and C get the same plasmid, and that's the plasmid that does not make the guide RNA. And so in these two, we do not see any cutting, right? That LAC-Z gene is still intact, giving us the blue colonies. Now, you'll notice that C has way more colonies and they appear to be smaller, and A has fewer colonies that appear to be bigger. These bacteria in particular are very density dependent with their size, right? And so just in general, the fewer colonies you have, the larger they'll be. The more colonies you have, the smaller they'll be, right? And that it really doesn't have any bearing on the experiment itself. This might just be because they pipetted a little bit too much, onto their plate or whatever. There can be a million reasons. We're not really looking at transformation efficiency in this lab. It's just looking at the outcome and the phenotype of the bacteria themselves. So don't worry about that too much. In our D plate, this is looking great. We have white colonies just like we would have expected. And our B plate, we have no colonies, just like we expected, right? And so that is exactly the kind of thing we would expect. This one, you can see that all those colonies kind of are about the same size, A, B, C, and D. Great. Here's another one. And I wanted to show you this one because someone sent this to me as a question. They said, everything looks right. A and C are blue, D is white, but B has this kind of blue haze on it. And if you look at this, there's actually that blue haze is appearing in other places as well, but there aren't any colonies growing on B. Now, what that is this blue pigment in the bacteria kind of washes out of the bacteria into the solution itself. And so that is what you're seeing on this B plate. It's the blue pigment from those starter bacteria, right? So it's not the bacteria themselves. Sometimes you see this, sometimes you don't see it at all. I just wanted to warn you that sometimes you might see this kind of faint wash of blue. Oftentimes they'll be kind of around the edge of the plate here. And that's just left over from those starter bacteria, the pigment in that starter bacteria. As long as you don't see colonies on here, you can assume that you're good to go. Now, what if you do see colonies in here? Well, bacteria are capable of fixing breaks on their own. And so that could be what happened there is you might get some, we call that non-homologous end-joining. And so that's another repair pathway that I'm not going to get into, but maybe they fixed it on their own. You would probably only see one colony, maybe. I've never seen this in the hundreds of time I've done this lab, but I have had some teachers that have seen this. And we have a lab that I was talking with Gary on that I think we're going to try to get on SLS2 is a genotyping extension lab. And so they're using PCR to see what's going on, right? So they take their colonies and they do PCR, amplify that DNA and see, does that bacteria have my stop codon in it that I added or not, right? And so that would be a great one to do PCR on. Okay, so those are the expected results. There's a little bit of troubleshooting here. So if you don't have any colonies on D or on other plates, that transformation efficiency might be low. Oftentimes this is, not every group in the class. It might be one or two groups that just have, maybe their technique wasn't perfect, right? And that's okay. I always have students save their A, B, C, and D tubes after the transformation. Just stick them stick them at room temperature somewhere. They're fine. And then if they don't see any colonies, have them restrike their pleats right over what they did before. Again, A on the A plate, B on the B plate. Those bacteria have now had more time at room temperature to divide, replicate, right? And so I bet you anything they're going to see colonies this time, right? So they might just need a little bit more time to grow. Okay, so don't throw those tubes away until you're completely done with the experiment. I say that of all of our biotech, all biotech labs, research in general. Don't throw things away until you're really done with everything. Okay. Again, colonies on B, we talked about that a little bit. And again, if you have that, entire class has that low transformation efficiency, just go to the whole leaving them in room temperature and plating them the next day. I, usually we're doing this in a rushed workshop usually. And so we typically get really good, really good results, even if we don't wait that whole 20 minutes, although the 20 minutes is best if you can wait that. If you can't wait that, just do it the next day. Just plate them the next day. Okay. So there's the review. I'm going to quickly go through that so we can get to the prep. Okay. So now this will be, slide deck will be given to you or available on the website, and you can click on here and that will go to our resources page, right? So you can find out more about CRISPR. There's a whole video on this, and that's me narrating and doing the lab so you can see exactly how this works. So it's about, I think, 20-30 minutes of me kind of walking through the lab and doing the student protocol. Okay, so that's handy on YouTube. There's a whole playlist on YouTube, and including that video right here. This is one where my colleague Damon is talking about some of the history of CRISPR. Here's one where I'm talking about the genotyping lab, which is that PCR lab, right? There's a webinar I presented that talks more about the history of CRISPR and what CRISPR is. And so all those resources are in that playlist, along with things that are not Bio-Rad, right? So there's an Amoeba Sisters video, there's TED Talks, all that good stuff on here too. So that's really handy. The instructor guide, you can download that, and that is free. You can look at it, now and see what you need, and the student guide as well. I did not talk about the student guide in there, in the back of it, there's a modeling piece that students can cut out these paper models and see how Cas9 cuts DNA. And if we have time at the end of this, I can go and show you what that looks like and how that works. But that's all in the student guide. There's also a capstone project at the end where they can do some bioinformatics. The bioinformatics are really great if you're introducing this because they're pretty simple, but they're going through the bioinformatics and thinking how they might address one of 3 different diseases. So one of those is cystic fibrosis. I'm just going to use that as an example. They would look at the gene responsible for that and figure out what region of that they would target with a single guide RNA. They would, write that sequence out, and then they're going to check and see if that has any off-site targets. In other words, does that sequence that they're looking to fix appear elsewhere in our genome? We don't want to be cutting something that, yes, it's, near the cystic fibrosis gene and we can fix that, but also it's cutting on a different chromosome entirely and messing things up, in an entirely different way. That's what we don't want. Those are off-site targets. And so they're using BLAST to search for those off-site targets and then making some recommendations about how that would work. So that's a really nice exercise if you're looking to do some bioinformatics and things like that. That's all free. You don't need to do the kit to do that, but you can download it. Okay, so what comes in the box? So here's a picture of the box contents. You get the E. coli, okay? And this will look very similar to the E. coli in the PILO kit. It is not. It has a different plasmid in there. This plasmid has one that is expressing, in particular, kanamycin, and we'll talk about that in a minute. So you cannot substitute just plain old E. coli, the HB101, or the K12 E. coli from PILO, because we need a different plasmid in that E. coli that they're starting with. You get those 2 different plasmids, the PLZ donor and the PLZ donor guide. You get Kix Mix, and this is a solution that has the kanamycin, the IPGG, and the XCal. You get the spectinomycin, which is the antibiotic. You get arabinose, which is that sugar, that pink dot that allows us to turn on the expression of those repair proteins. You get LB agar and LB broth, loops, petri dishes, microcentrifuge tubes, transformation solution, and this kind of easy start guide, which is, here's what the kit is and here's where to store everything and all that good stuff, and the instructor answer guide. So that is what is printed in the box. The instructor answer guide is really important for you to hang on to, because that's where all the answers are from the student manual. The student manual does not contain the answers for obvious reasons. They are in this printed answer guide, okay? So here is all of that in this format because I like knowing what things look like, right? So it tells you the E. coli is in a small brown glass vial, right? And the transformations in a clear plastic bottle. And those are all in a baggie that gets stored in the freezer. And it tells you on there to store at -20 Celsius. And of course, in the US, we are not good at Celsius. So people constantly store things at the wrong temperature. So I've taken to just saying freezer, fridge, or room temperature in order to circumvent that a little bit. You guys all know what Celsius is. So -20 is quite cold, right? So it's freezer. And then the rest of the components can just remain in the box at room temperature. Okay. And of course, you'll need to thaw these before we go to prep. Now, other things that you'll need, the P1000 micropipette and tips, you'll need one for prep. The students could use them as well, but they can use the disposable transfer pipettes. Okay, so you'll need a balance and an incubator, optional. They could do room temperature. You need either an autoclave or a microwave. I prep this in my kitchen. I don't have an autoclave, obviously, so I use a microwave, and I'm going to show you how to do it with that. You'll need 500 ml flask or bottles. And actually, I should say this with a caveat. If you have an autoclave, you can use 1 liter bottles, okay? And that's actually what it calls for. I find that most teachers don't have access to an autoclave, so I'm going to show you how to do it with a microwave. So we're going to talk about the 500 ml flask. Centrifuge, optional kind of. You do need a liter flask. It's not going to go in the microwave. It's basically just for mixing. Graduated cylinders, distilled or deionized water. Whatever you see deionized water, you can use distilled, okay? Permanent marking pen, like a Sharpie, aluminum foil, bleach to clean up, lab tape, that kind of things. Okay. The students will also need those P200s and P20s. Again, we talked about how you can use disposable pipettes, or you can pre-aliquot the plasmid so that they don't necessarily need these. They're optional, but recommended. You'll need a water bath with a thermometer, right? To tell you how hot it is. We want 60 degrees Celsius. I find it nice to have things in a rack for their tubes. Foam floats if you're using a water bath. I use the crushed ice water bath. Okay. And so I don't have access to like the nice nugget ice that you can get in a lot of labs. If you don't have that, that's fine. Cubed ice is fine. Just make an ice water bath. We want to make sure that the tubes are touching something really cold when we're doing that transformation step. And so if you have big cubes of ice and you're putting your tubes in there, they might be just touching air, right? And so putting them in an ice water bath makes sure that the transfer of cold gets into that tube really well. And the scissors are just for that paper modeling piece if you do that. Okay, so the prep timeline. Now, there's two different timelines here, one with an incubator and one without. So let's look at the one with the incubator first of all. So the first step, you're going to pour those plates. And this is at minimum, right? This is I typically show you guys the last possible moment to do things because that's just how I operate, right? You can always do this earlier. This could happen up to a month beforehand, all right? But at least 3 days before the lab, you're going to want to pour those plates. And they need time to sit at room temperature. They'll sit with the lid up, so not upside down. They'll sit with the lid up for 2 days at room temperature. Okay, they need to be in the dark, like I said before. You're going to need to rehydrate. That bacteria comes dried up, it comes lyophilized or dried up. And so that needs a day in the incubator. And so you'll do that at least 2 days before the lab. You're going to want to rehydrate that E. coli. Then you'll make your starter plates the day before the lab, right? And those need to incubate for 24 hours. Now, this is kind of crucial because we want good, fresh starter plates. If you are incubating, if you can't, you want to do the lab on Monday, let's say, for example, and you don't want to go to school on Sunday to streak starter plates, go ahead and streak those starter plates on Friday, but leave them at room temperature. Don't put them in the incubator. They'll be too overgrown and old to use by Monday. Okay. So this timing is kind of dependent on, we want to make sure they're good and fresh, either 24 hours at 37, or as you'll see in the next slide, 3 days at room temp. Dispensing solutions, this can happen whenever. Typically, people do it the day before the lab, right? You could do it a month in advance. It doesn't matter there. And then you'll set up your lab stations the day before the lab or the day of the lab. That would be fine too. Okay. Now, without an incubator, you're going to shift things back about 2 days. Okay. So we're going to prepare plates 5 days before the lab and let them dry for 2 days. You're going to rehydrate the E. coli 4 days before the lab. Okay. Let go overnight. And then you can streak those starter plates. So starter plates are going to need to sit at room temperature for 3 days. Okay. So that's why you see this longer tail here, right? And then dispensing solutions and setting up lab stations whenever you want to do it. Okay. So either way works. This for me all comes down to when I want to do stuff and when I have time to prep, right? I have an incubator, but I don't always use it. You can pour those plates, I would say 2 weeks to a month in advance, a month for sure if you're using an autoclave. I only have a microwave, like I said, and I regularly do a month in advance just because, or more typically what it is that I've done one workshop and I have leftover plates and I'm doing another workshop and I just use the old plates, right? And so that works fine. You're going to stack them 20 plates high, slip that original plastic sleeve over the stack, okay? And flip the entire stack over and then tape the top closed and store it in the fridge auger side up. Okay. So when you get the plates, don't just cut the end of the sleeve off neatly at one end, and that way you can use it just to store your plates in. And then, of course, just check for contamination. If you see any kind of fungal growth or bacterial growth on the plates, just toss that one plate. Usually it might be one or two, right? It won't be the whole sleeve unless something's gone terribly wrong with your prep. Not, that happens. So that's okay. So the solutions that you're aliquoting out, like the LB and transformation solution, that can happen way far in advance. Just keep them in the fridge. Glassware. I use these 500 ml media bottles. These are, Pyrex or Corning makes the other, Amazon honestly has other vendors and they work just fine. You want to make sure that they're not going to explode when you put them in the microwave. So this borosilicate glass, they have a plastic lid that's also microwave safe. Just put that on and then back off of it to leave it loose while you're microwaving. The other option is to use a 500 ml flask. And I like to turn a 50 ml flask over the top inverted and that kind of captures the condensation so it drips back into this flask, okay? And you can see here, those are touching the top of my microwave. So this is not my ideal setup, but I do these 500 ml media bottles. Again, if you have a larger microwave or you have an autoclave, everything will be in a 1 liter bottle and that'll be fine. Okay, so prepare the flask. You're going to need 1 Kix flask and 2 Kix SPT flask. You can just label those. And then you'll need another 1 liter flask just for mixing things up. You're going to add water, 3 mils to the arabinose and 500 microliters to the spectinomycin. Arabinose will take you quite a bit to go into solution, at least 10 minutes. And so don't despair if you're looking at it and it's still not going into solution. We're doing this now so that it has plenty of time. Okay, the spectinomycin will go into solution immediately. Okay. Now we're going to work with the Kix mix, okay? And so you're going to add 700 mils to that 1 liter flask, 700 mils of water to that 1 liter flask. And then the Kix mix is this white flaky powder. And so we're going to need to kind of wash it out of the vial and put it into our, I'm sorry, that should not say Kix SPT. This should be into our 1 liter flask here, okay? So you're going to add 3 mils of water to the Kix mix and wash it out into that 1 liter flask. And you're going to do that 3 times total, okay? It will not really dissolve until it gets microwaved. And so that's a little concerning to some people. I'm going to show you what it looks like. This Kix mix has in it kanamycin, and that's an antibiotic, IPTG, and Excal. And so those are hazardous if inhaled. So you don't want to, pour the powder into this bottle until you've added water to it first, right? And so I just want to show you, if you look on the right here, you can kind of see those white flakes. That's all the Kix mix, and it will not go into solution until you microwave it. You do want to make sure that there's not a bunch of white flakes inside your bottle. This is a kind of a hard picture to tell, but I've washed it out 3 times, and there's still always going to be a little bit of white residue in here. We just want to make sure there's not a lot of white on the inside of the bottle here. Okay, so rinse that out as well as you can. Okay, and stick that in your 1 liter flask. Now you've got your water and you've got your Kix mix in this flask. You're going to give it a good swirl, and then immediately put 200 milliliters in your flask that you've labeled Kix, and then 250 milliliters into the Kix SBT flask. I do not use a cylinder for this, and I know that's probably, sacrilege. We are just trying to get this in here, and if you go and take the time to measure it carefully with a cylinder, that Kix powder is going to sink to the bottom, and you're not going to disperse it well. Just use the markings on the side of the flask, get 200 into here-ish, and get equal amounts into here, right? That's all we're going for here. It's fine. Should you use a cylinder? Probably. In this case, though, it's going to make it where your Kix isn't distributed evenly. So that's kind of the goal here. So 200 in the Kix flask and 250 each into the Kix SBT flask. Okay. Now we're going to add the LB agar. So this is the same kind of agar that we would use in Piglo or something similar. And you're going to add 7 grams to the Kix, 9 grams into each of the Kix SBT. Okay. Just weigh that out. Give them a good swirl, and then microwave until it's boiling at least 3 times. I also have taken once I bring it to a boil, I'll lower the power on the microwave and let it simmer in there for 2 minutes, okay? We want to make sure there's no kind of swirlies of undissolved agar left in there. If you see that, then your plates won't set up. So this should look very clear all the way through. You might see bubbles, but you will not see any, kind of swirlies, clumps of undissolved agar or anything else in there. Then you're going to prepare your plates. Okay, so we're going to label them. Now you can label 8 plates X, 8 plates X era, 8 plates X SPT on the bottom of the plate, right? On the agar, what will become the agar side. This is way too much labeling for me. So here's what I do. I use a stripe, and I put a stripe down. I just take a Sharpie. I have my plates stacked up, and I take a Sharpie and draw it down the line here. And I tell my students, okay, the one with 1 stripe is X. You take your plates with 1 stripe, flip them over and write X on the bottom. They do the labeling for me, okay? The 2 stripes are X era. So find your plate with 2 stripes, flip it over and write X/era on the bottom of that plate. And then other plates that don't have any stripes are all XSPT, and they can label those as well. We'll get 4 of those, right? And so that's how I label my plates. I make the students do it because this is a lot of labeling, right? So that's what you're going to have there. I wanted to show you how I pour my plates. This is sometimes a revelation to people. This is just how I learned in grad school. So I usually stack them up in groups of 4 or 5. This is my boiled agar. It's now cool enough for me to handle, right? That's the temperature it needs to be. Stack it up, remove starting at the bottom, I remove the other Petri dishes, the lid of that bottom one plus the other Petri dishes, and I just work my way up to the top, okay? And I'm not measuring here. I just pour until it covers the bottom. And then, at the end, I usually give it a little swirl just to make sure, just in case it didn't cover the bottom of all of them. But that makes things go really, really quickly. Okay. So we're going to pour the plates. So you'll wait until you can hold the bottle. You don't want to wait too long. It's kind of a fine line. So 50 degrees Celsius, you can stick it in a water bath and that'll get it to that temperature and hold it there better. I don't have, like I said, I'm not going to pull out the water bath in my kitchen, but I just wait until I can hold it. And we're going to pour 8 of the Kix plates. That's just straight from the microwave from that 200 ml flask, right? And then we're going to add to that vial, we will add arabinose. Okay, so 1 ml of arabinose into that Kix vial. And now we've made Kix era or X era, right? And so you'll notice that we go from Kix, which is that Kix mix kanamycin, IPTG, and XCal, to labeling it just IX, right, for IPTG and XCal. And it's because this kanamycin is an antibiotic just to prevent other growth of other bacteria. Remember the E. coli that we ship you has a kanamycin resistant plasmid in it. It's not really integral to the lab and we don't want to confuse students with these 2 different antibiotics. So we just drop the K out for student facing supplies like these X era plates. So if you're wondering why we don't call these Kix era, that's why. Okay. So we're going to add the arabinose to the remainder of our Kix augers, give it a good swirl, and pour 8 of the X era plates. Okay, so we have the X plates here and the X era plates here. These are where are going to become the starter plates. Now we have 2 more vials of this Kix that we've labeled Kix SPT, and those have 250 microliters of agar in them each. We're going to add 250 microliters of spectinomycin to each of those 2 vials. So we have 250 into one of the 250 ml flask and 250 microliters into the other 250 ml flask. Give them a swirl and pour 40 of those plates, XSPT plates. This is the part that's kind of crucial to let it go, make sure it's at 50, 55 degrees because spectinomycin will be degraded at higher temperatures. Okay. So that's, it is a little bit of a timing trick because if you let it go too long at room temperature, it's going to cool so much that it will get clumpy or that you can't pour plates from it. These have to be stored in the dark. I usually let them sit. It's not imperative that they get stored in the dark right away. Let them solidify, firm up, give them an hour or so, and that's fine. Just, I have them on a cafeteria tray basically. And then I just stick them back in the kit box and let them sit, carefully out of the way until we're ready to use them. They need to be at room temperature, lid side up, not upside down, for 2 days in the dark. And that will allow that agar to dry up a little bit. We don't want these plates super moist because we want the bacteria to to get into the agar. We don't want it just sitting on top. And so that 2 days of letting them cure at room temperature is really important. After the 2 days, you can use them straight from room temperature, or you can put them in the fridge. And if you put them in the fridge, you're just going to stack them 20 plates high, put that original sleeve back on top, flip the whole stack over and add tape at the top. And then I like to wrap it in aluminum foil and stick them in the fridge. I don't have room to stick mine in the fridge upside down. I just lay them on their sides and they're fine. As long as they've solidified for 2 days at room temperature is fine. Okay, so that's our plates. Our plates are done. Now, we're going to need to also rehydrate the E. coli. This can happen the same day you do the plates if you want, or you could do it, the following day. But you need to make some LB broth. We give you a capsule. We'll just add that into a bottle with water, microwave that, and let it boil 3 times, and then let it cool down to room temperature. And then you're going to use that to rehydrate your E. coli. Okay, so the E. coli gets 250 microliters of the broth. That E. coli will need to be incubated either overnight at 37. You could do it 8 hours. I usually just find that I'm usually doing it overnight, so at least 8 hours at 37, or room temperature. 24 hours at room temperature is sufficient too. Once you've added the LB broth into here, we usually like to have you boil the rest of the LB broth again, just to make sure you didn't accidentally contaminate it, right? Because we're also going to use this for students. Okay. Now, once you have your plates that have been cured for 2 days at room temperature and your E. coli has grown overnight, you're ready to streak the starter plates. And you're going to streak 8, you're going to use E. coli on all of them, and you're going to streak 8 of the X plates and 8 of the Xera plates. We like to do this 4-phase streaking like so, and that will give you hopefully some nice colonies in the middle of the plate. And those need to be incubated for sure at least 24 hours at 37 or 72 hours at room temperature. Okay, so 3 days at room temperature. And again, like I said, keep those in the dark. Okay. They'll need to get solutions here. So the transformation solution, the LB, and the 2 plasmids. The transformation solution in the LB, you can use a regular pipette, a P1000 for this. I prefer to just use a disposable pipette, and I just draw up a little bit more than 1 milliliter and dispense them into the tubes that way. I just find it's much easier. You're not having to change tips. You can get you can get that into the bottle easier. I don't know. Just for me, I prefer to do it that way. You could, of course, use a pipette or however you would like to do a little bit more than a milliliter. They need exactly 1 milliliter, so you want to just give them a little bit of extra. So transformation into the TS tube, LB into the LB tube. The plasmids, if they have P20 micropipettes, you're going to aliquot 25 microliters into the PD tube. They'll use 10 of that to go into tube A and 10 into tube C. So they should have 5 microliters left, right? So just to account for pipetting errors. And then 25 microliters into the PDG tube. And again, they'll use 10 for tube or for tube B and 10 for tube D. Again, if they don't have P20s or you don't have enough, you can go ahead and just grab some more tubes and give them PD into tubes labeled A and C and PDG into tubes labeled B and D. And you would just put 10 microliters in at that point. Okay, so this is the step that you can change if you don't have your students using micropipettes. Okay. And then all of those solutions get stored in the fridge until they're ready to use. And again, this stuff can be aliquoted out well in advance. It could be, several weeks in advance if you wanted to. The students are going to have this workstation. So the 2 starter plates, X and Xera, they'll have the 4 X SPT plates. So again, just as a reminder, the X plate is the one that does not have a arabinose, and so the repair proteins are not turned on. The Xera plate does have a arabinose, and the repair proteins are turned on in that one. The X SPT plates are X plus the spectinomycin antibiotic to select for our bacteria. They'll get LB broth, transformation solution, the 2 plasmids. The P1000, P200, and P20, really these are all optional, but recommended. So you could use disposable pipettes here if you want. They'll need microtubes, 4 of them that they'll label A, B, C, and D. They'll need loops. You'll need to have an ice water bath available at each station, a Sharpie, a waste cup. I like to have a tube rack and a foam float. And then somewhere in the classroom, you'll need to have the water bath set up and an incubator, that's optional as well, and lab tape available for them to use to tape their plates together. That could be masking tape or painter's tape or whatever if you don't have lab tape. Okay, I think we've talked about this. Yes, I've got this here again. I think that is it. The last thing I wanted to say is we do have a new lab. I think this is coming online on SLS's site soon, but if it's not there already, and this is about quorum sensing and bacterial communication. So if you have any questions about that, I'm happy to answer those. If you have any questions about CRISPR or how it works or anything that we've talked about, now's your time. Let me know, and I'm happy to answer questions. I have a whole other slide deck on kind of how CRISPR works. It's got the modeling information. I'm happy to go over any of that if anyone's interested. So you guys just let me know.